everyone's, can you mute everyone, Yunus, and then,
Yes.
right. Aisha, you have to unmute also.
There it is. Okay, should we get started?
I think we can, yes.
All right, so Eunice, if you want to start recording anytime soon, that would be great.
So who is recording? Just to.
I already start recording on the cloud.
Okay, okay.
Okay, wonderful. So I'll get started. So again, hello and welcome to another lecture in this series of lectures that we have been doing on the topics of medical genetics. And some of our co-sponsors are, of course, the Pakistani Society of Medical Genetics, the University of Health Sciences, Lahore, as well as Jinnah Sindh Medical University Alumni Association of the North America.
Proper medical care in this country.
About America? In the beginning?
Yeah, they said.
Okay, so next slide, please. Okay, so today's lecturer is going to be Dr. Rizwan Naeem. Dr. Naeem is a professor of pathology at Albert Einstein College of Medicine and serves as a founding program director for Laboratory Genetics Fellowship Program. He is a graduate of Sindh Medical College and executive director of Sindh Medical College Alumni. Currently, he's serving as a president-elect of the Council of Program Directors at Association of Molecular Pathologists, and we're very delighted to have him give our lecture today. It's going to be a wonderful presentation, I can already tell, and I am very excited to be here, as I am sure that all of you are as well. So next slide, please. I would like to go over some disclosures so he does not have any relevant or material financial interest that relates to this talk.
You can, I didn't put the slide for
okay.
house.
Right. So the housekeeping rules are as such. We would like everyone to stay muted for out of respect for Dr. Rizwan Naeem and out of respect for everybody else on the call, so we can all hear him clearly without interruptions. And of course, we would like to see a lot of questions, a lot of discussions that actually happen on the chat box. So please feel free to put all of your questions in the chat box. And Maila Ashfaq, who's our genetic counselor, and myself will be monitoring the chat boxes. We will hold on to those questions until the very end, and we will post those questions to Dr. Naeem. If we are able to answer those questions directly, we will post the answers directly in the chat boxes. And lastly, please feel free to take a look at the form that we have for you to fill out. That's the only way we know how to keep track of participations and interest in these lecture series and a certificate program in the near or distant future. So please take the time to click on the link. We will be sharing that link periodically during the lecture and towards the very end as well. So please feel free to take a look at that form. It's a Google form and fill it out. And so that's it. Without any further ado and taking any more time, I'll hand it over to Dr. Rizwan Naeem. Take it away.
Thank you, Aisha. And Aisha is the heart and soul of this whole process. And thank you, Yunus, for providing all the material support and support from our alumni, Sindh Medical College alumni. And so one thing I wanted to add, this filling out these forms are so important that if we have your email, we will keep informing you what we are doing. And please also visit our website. It's called www.psmg.org, Pakistan Society of Medical Genetics.org. So with that said, I want to just briefly update you that what we are going to cover today. So what I want to say that this will be an overview. Lab genetic is such a broad topic that cannot be covered in 45 minutes we have. So it will be just an overview and why we need it and how we do it. Basic understanding, it will cover basic understanding of what is much more complex topic. And intentionally, it is some of the things are glosses over finer detail in favor of simplicity. Often cartoonishly simple understanding of the concept. Sorry. So what I know we will not cover or provide a comprehensive review of all the application of genetic testing, an understanding of actual steps and effort used to perform genetic testing in the laboratory. So just to start with, I will, I promise you during this talk, I will take you to the moon also for a change, and give you concept that that what lab people do in making medical decisions, in making diagnosis, and patients' medical records. So it is a fact for now that 70% of medical decisions are made by knowing the laboratory diagnosis. And 70% of patients' medical record is actually lab data. So that's how important laboratory medical people are, which includes genetic laboratories. So I also like to first clear two very basic concepts. I will try to. One is, a lot of people think, and especially medical people think, that genetic disorders are very uncommon, which I like to provide some data that it is actually very common comparatively. So just to give you some example, that many of us have seen miscarriages in our family, our relatives, our friends, and so on. So 50 to 60% of miscarriages in US or anywhere in the world actually are due to genetic reasons. And we can find those genetic reasons and try to prevent it. And almost 0.5% of the live-born children have genetic issues, which can be worked with laboratory genetics to see what they are and what need to be done with them. So over 200 pediatric diseases are due to extra or missing chromosome or piece of a chromosome, which delineate to multiple major or minor anomalies with mental disability and syndromic findings. Point mutation and exon level alteration in human genome can cause single gene disorders or syndromic disorders. So with that said, I want to cut the end part and say genetic disorders are very common. And I hope you agree. So let's see what's happening in Pakistan. So I was in Pakistan last year in February, and there was a conference, and Dr. Huma was the head of that conference. Her proceeding came out in one of the newspaper, Dawn, and this is what she said. Genetic diseases are in most cases serious, chronic, and debilitating and life-threatening, often requiring long and specialized treatment. Largely neglected, genetic diseases are responsible for 50% of death in children before the age of 1 year in Pakistan. That is pretty significant. Think about it. And some of these can be preventable. I'll explain you how, by the laboratory diagnosis. Responsible for, and genetic disorders are responsible for significant handicap in older children also who have muscular or CNS diseases. She continued that after infectious diseases and malnutrition, which is of course is a problem in all the third world countries. So after infectious disease and malnutrition, genetic diseases are the biggest cause of morbidity and mortality in children in Pakistan. And a very high prevalence of inherited or genetic diseases in children. And we know why, because like many other segments of population, we do a lot of inbreeding in Pakistan also, marrying to their own family members, marrying to the cousins, and same people which are inbred in areas very limited to their own. Very other good example is Ashkenazi Jewish population for example, with we see a lot of genetic diseases, and that's why we believe that there in Pakistan also there's a lot of inbreeding and genetic diseases, autosomal inherited type especially. Physician caring, but the drawback is, and that's why people like Aisha and Myla and Ijaz and myself, we started the society with the help of other helping hands from Pakistan and US, that we started Pakistan Society of Medical Genomics or Genetics. And the reason is that it is very, and there are multiple papers about this also. Myla actually has written a paper on this also. Physician caring for children many times do not have the acquired knowledge or expertise to guide families with such disorders. So that's why we needed genetic counseling. That's why we need someone to explain to patient or to physician that what is important and how we can deal with it. So again, bringing the same point with some graphic and data that nearly 10,000 conditions, medical conditions, can be identified using a genetic test. And that number is keep increasing. This data only shows till 2017, and this is every day there is a new genetic disorder, new syndrome, which is caused by alteration in our genomics, by DNA or by chromosome or by expression analysis. So this is a data from one of the private lab in US. They were just calculating, this is a lab which do a lot of different testing, but they provided this data on the website that how they are selling DNA-based testing. So look at this data. So it's almost doubles every year. And I believe that this trend is continued to go up and up and high and high. And so that's how common genetic testing is all over the world. This is data is from US, but I'm sure this is common everywhere. So I hope by these examples, what I convinced that genetic disorders are very common and cause a lot of disease burden in society, especially in third world. And there's significant increase and significant percentage of increase of morbidity in children which are 1 year and younger, and significant more disability issues in people in people in middle age and high, older ages. So that is very common, and that need to be dealt with. And one of the reasons we will provide that how we deal with. So now clear up the second, what you call misconception. When you go to a physician and say, okay, you can do a genetic test, there is a concept or misconcept, I would say, that they are very expensive. And yes, they may be relatively expensive to a certain degree, but I'll show you examples that how this cost is coming down and in a very, very significant manner. So for example, when first human genome was cloned, it cost US $2.7 billion. And now with technology advancement within 10, 15 years, 15, 20 years of technical advancement, with next generation technology, a human genome for sure can be sequenced for less than $10,000. And I believe, and many of us who are geneticists believe, that in foreseeable future, the goal is to make it $1,000 human genome sequencing, which we are going very rapidly towards that cost basis. And this is again to prove my point that this is data from National Human Genome Research Institute. As you can see, the cost of human genome sequence has plummeted, and genetic testing has plummeted in last many years, and it's keep going down. the cost of genetic testing is keep going down. So what I mean is now that it is not, hopefully it's not that expensive now. And I believe that it will be much less expensive as we move forward. So now let's come back to our topic again. And that's where I'll try to take you to the moon. Just follow me and take the ride together. Why we need it, genetic testing or genomic, laboratory genomics, and how we do it. So whenever you are looking at genetic testing, just keep these two things in mind: why and how. How to decide what is the right test and methodology? And that's a very complicated question. And I'll try to simplify as much as I can. And the next part is there any benefit? But when you're talking about genetic testing, is it really benefit individual or in community in general? And if time permits, that's my last topic of my talk, that if time permits, we will talk about that. It's not only very beneficial to individual who is testing, but it's also very beneficial for the society you live in. So when we go to genetic or laboratory genetics and what to do and how to do, I would like you to think about this. Hammer, right? We all have seen it. We all have used it somewhere and somehow. But you also know that one hammer cannot be used for everything, right? So these are different type of hammers. Same way, genetic technology, genetic testing is very different for very different users. So you need to know what you are using for to understand what type of hammer you need to use to tackle that genetic issue. So these are types of genetic testing, for example, why we do it. And this is, again, that's not a complete list. I know I'm looking at it and I know I'm missing, for example, forensic testing, right? So there are many, and microbiological based testing for laboratory genetics or gene sequencing. And very good example I'll show you. So so you have to understand that why you need this genetic testing, and then understand what the technology is used, how the best technology can be used. So now let's go to the moon, right? So suppose you are an astronaut and you are and this is explained how we do it, right? So you are an astronaut and you are at in some of the big microscope looking at the moon. And looking at the moon, you see that moon shines up at the night. Sometimes it's bigger, sometimes smaller. And also there is an old woman, jo chakki chalate, sitting there and doing that. And that is for someone who really very good in looking at the moon and microscopes. So, but what I meant is this, that genetic testing is also something like that. So when you look at genome by very far away, you will see the first picture. Let me see if I can get the marker. Yes, you will see this type of thing. And believe me, when I started my genetics career, I was the one who was counting chromosomes. And by this type of technology, where there are no bands on chromosome and just counting dark chromosomes, geneticists discovered what is Down syndrome is. Geneticists discovered what is Turner syndrome is. Down syndrome is when you have an extra chromosome 21, and Turner is when you are missing one chromosome X. So you can see, but this is a good technology. You're looking at the whole genome, but your resolution is very low and you're only counting chromosome, which now is almost obsolete. So then people move on to see how we can go close to the moon. You are an astronaut, remember. So you want to go as close as you can and want to see what's happening on moon. So you said that I'm going to go, when I grow up, I'm going to go and have a ship or rocket and go to the space. So you travel to space now. And from space, you start looking at human genome, which is much higher resolution. So now what you're seeing is this one chromosome can be divided into light and dark regions. We call it cytogenetic bands. And that technology actually is still in use and one of the most common technology we have used other than NGS for now. And that has many big applications, many different applications. And geneticists like us got very, very excited, and they discovered many different genetic diseases. For example, Philadelphia chromosome, when you see that Philadelphia chromosome in CML actually is a combination of chromosome 9 and 22, which is a diagnostic mark for CML. Same thing is many syndromes, deletion syndrome, very common DiGeorge syndrome, very common telomeric deletion syndrome, very common. So there are a lot of different syndromes, which just by looking at these bands, people realized, and you see how beneficial it is to look at the whole genome, but also go to the space for looking at moon rather than looking from the Earth. So that technology is very useful. Pakistan also has this technology. Places like National Institute of Child Health and Karachi, in Lahore, many places, Armed Forces Institute of Pathology, Aga Khan University, and many other places have this technology. But again, you are not looking at moon, and you cannot have access to look at each stone and try to analyze that stone at moon. But you're looking at the whole moon, but at much closer place than the Earth. So, but then we move on to microarray and sequencing, and which means that you really travel to the moon and you are landed on the moon and you start looking at each stone and each places together. So let's move on, say how we move next. And I'll give you example how this technology evolves. So this is, as I mentioned, this is chromosome analysis, very significant, good banding. As you can see, the pair of chromosomes are matching each other, and we identify that this is chromosome 1, and this is chromosome X, and this is chromosome 17. So once you keep looking at these, this is called cytogenetic or chromosome analysis. And the most common application of this in early 90s and late 90s and early 2000s is chromosome analysis for pregnant women. So situation is that if a pregnant woman comes to the clinic and have an abnormal ultrasound, if you have an abnormal ultrasound, what are your choices? You want to know if the chromosome, if you have any syndromic changes, if there is any trisomy or monosomy or big chunk of DNA missing someplace, and you are still remember, you're still in space, you're not landed in moon yet. So what do you do? So this was a real case in my lab, and we saw that this, rather than 46 chromosomes, there were 47 chromosomes. And we have this extra chromosome, we call it a marker chromosome. So I'm sure many of you will realize where this is coming from, right? I don't. So for example, there is no way, I'm in genetics for almost 30 years, but I cannot figure out where this extra chromosome is coming from. And I'm already on a space, so I should be looking at it carefully. This can be from chromosome 2 or 1, or maybe from 22. Look at this. I mean, these bands are light band and dark band, and the patient is pregnant, almost 16, 18 weeks pregnant, and then she needs to know what it is before she decides to continue with the pregnancy or terminate it or whatever to do with that. So then geneticists keep working in laboratory to see how to identify. So what my point is here, the laboratory genetics, cytogenetic is a very good technology. It gives the picture, whole genome picture of human genome, but also is very low resolution. You are still in space. You cannot land on the moon to start analyzing things in detail. And that's a shortcoming of cytogenetics, in addition to many good use for cytogenetics. So then we move on in laboratory. We wanted to see things at much more detail and much higher resolution. And then this technology comes in. It's called FISH. And I'll tell you why it is called FISH. It's not because it's fishing, but it is like fishing, but it's called fluorescent in situ hybridization. Right? So what geneticists or laboratorian like you all come with an idea that if you have a double-stranded DNA and you denature it, you can denature it with heat, for example, or some chemicals. So it becomes single-stranded. And then if you make a laboratory-made probe, which is very specific to the sequence you are looking for or fishing for, and you put that probe with a color and tag it with a color, and then have an abundance and hybridize it to the single-stranded DNA. So you can see these two sequences are in 1-to-1 copy number, but if you have a probe which is colored and much abundance, that will have a higher chance to hybridize with your target chromosome or piece of the chromosome. And this is how they do. It hybridizes with your target you're looking for, and then you wash away the rest of the probes, extra or additional probe. And then you see on an interface cells or metaphase cells, doesn't matter, you see if your probe, which was here, hybridizing to your target or not. And that's what fishing is. So it is very, very high resolution, right? So this probe can be up to 200 kb or 150 kb, depending on how your microscopes are and how well hybridization is. If you have 200 kb or 150 kb, you should be able to know if that sequence is present in your cells or not present in your cell, or what is the position of that hybridizing probe. So that is a much, much bigger advancement in human genetics, laboratory genetics. And now what I mean is, now from Earth, you went to space with banding, and now you are in the orbit of moon. And you can see at a much higher, but you still do not have technology to land on the moon, right? So we are much, much hybrid, a higher resolution. The advantage of fluorescent in situ hybridization is that it has very high resolution, comparatively speaking. But the drawback is it is not comprehensive. You're not looking at the whole chromosome. You're just looking at a very small piece of DNA and seeing it, how it is present or not, or in what orientation it is present. Sometime if you use two probes to see the orientation. So this is some examples of fluorescent hybridization. So very, very small deletions, for example, in chromosome 13 and chromosome 21q or trisomy 18, you do not have to do chromosome analysis, because remember, when you do chromosome, you have to culture the cells, you have to make preparations, slide prepare, you have to band them with certain chemicals, and then look at the whole genome and align them. So you don't need to do that anymore, and you can just use interface cells. But interface cells mean you can also use dead cells, right? So if you have a miscarriage, say 5 years ago, 4 years ago, and you did not know that if it was a trisomy 18 or not, you can still use that paraffin embedded tissue and hybridize it. It is very, very commonly used in cancer. For example, many paraffin embedded or old slides can be used for this fish analysis. But the thing to remember is that you are in the orbit of moon, but you're not landed in moon. You can see things if you know what you're looking at, but you cannot see what you're not looking at. So for example, in the example I gave you about cytogenetics was this, that you look at whole thing. We are not expecting, we were not fishing for that. We didn't know if this chromosome is present or not, but that's the advantage of looking at whole genome technology versus looking at very targeted fish-based approach. So people have very much modified fish probes in many different ways. There was a time where every journal cover page is like, with this colorful photos, CMYC, MYC genes, PAX5 gene, BCR-able translocation, and sky karyotyping that we can color each chromosome with different colors, and so on and so on. And telomere, for example. So this is very beautiful and very good technology if you know what your target is and what you're targeting for. And that is very easy to use, as I explained in the graphic, that denaturation, hybridization, and looking at cells. But let's come back to that journey we started. So we are now, if we want to do fish, tell me, where do you want to go? Which chromosome, which target you want to use? I know many of you might know it, but I don't, right? I cannot start fishing for one or the other chromosome. And it may take, and fish technology may take a day or two to get an answer. So if you have 22 chromosomes, you might be taking 44 days to analyze this if you do it. And then fish for what? It is not a whole chromosome. You cannot fish for one whole chromosome. You only fish for part of the chromosome. So this is a problem. We are still not to resolve this unless you land on the moon, or maybe before that, maybe. So then geneticists or lab people came up with a new technology. So if you remember what I mentioned, that chromosome analysis is a very good technology, and it gives you the whole genome perspective. You're looking at the whole genome. And FISH is a very good technology and a high resolution technology where you're looking at your target presence. Here it is present, here it is not present, right? So that is the difference between whole genome perspective and increased resolution. You are close to moon, but you still, we cannot look at the whole moon. So then when human genome was cloned or sequenced, for example, then what we did is we cut small pieces of human genome. So you take a human genome, human cells, and use restriction enzyme, for example, or shredding, we call it. You can shred it into very, very small pieces of DNA, which you can target where you want to target, like fish. So for example, you can have this, these are computer slides actually, right? Computer slides, and you implant or plant your pieces of DNA in whatever fashion you want. And then hybridize your test DNA, I'll show you another example, to this probes. If you have equal amount of target and test, the color will be yellow, because one color you're using is red, one color you're using is yellow. So if everything is equal, so it will be all yellow. Very simple. But if something is more or less, color will be red or green. And I'll show you some examples. And that's what it is called, genomic DNA as analyzable substrate and automation. So this is called genome microarray or array CGH or microarray CGH, many different ways. And that technology is still very active. I don't know if anyone in Pakistan is using it, but that's a technology commonly used for children with unknown medical conditions, and many use in cancer. But this has also its limitation. I'll show you what. So overview of array CGH technology, as I mentioned, This is just a graph saying that you have your pieces of DNA and you hybridize, you place your piece of DNA into a slide, glass slide. And then you have a test DNA and you have a reference DNA. Reference DNA means there is no such thing in DNA which is called normal or abnormal, right? So it is a reference DNA and a tumor DNA. So you color one with red, one with green, and then hybridize in equal amount to the slide. When that happens, as I mentioned, the yellow parts are showing that both reference and green and yellow, green and red are in same amount, so it will show a yellow color. But if as you are missing one or addition of another one, the color will be different. And this is how the karyogram will look like. So this is again, you're still not at the moon, but you're very close. You have a high resolution like fish, and you have a whole genome perspective like chromosome analysis. And that control and DNA test a purified label with fluorescent dye and hybridized to array containing SNP or oligonucleotide probes and span the whole genome. So you have a whole genome perspective and very high resolution. So now coming back to the same case, right? So now we have developed array CGH, and we thought that we can use this as a test DNA and use another reference DNA, which is a supposedly no gain or loss of human DNA. And then we tried that. So that was a de novo marker chromosome. And what we realized, this is a piece of chromosome 15 and 16. How that can happen? And that can happen because if one of the parent is a balanced translocation carrier of 15 and 16, chromosome 15 and 16, and you can see the same marker chromosome here. And we figured out within a few days that is actually and that technology was just starting at that time. I think it was 2001 and 2002. So that's how we resolved this case, and that's how the technology evolved. And so the disadvantage of this technology is this will not detect balanced chromosomal changes because you are only looking at if there is any imbalance, presence or absence of extra or loss of chromosomal material. And the resolution can be as low as 100 kb. So it's a great technology, but not complete technology because you're not looking at the whole genome. Also balance changes. So what are the common indications of CMA or chromosomal microarray? Multiple congenital anomalies. So you don't know where to start, what to do. Dysmorphic features, intellectual disability and developmental delay, autism spectrum disorders, and the cancer and oncology has its own separate world where microarray can be used and is being used. So in genomic or inherited conditions, for example, or constitutional changes in genome, 20% of the yield in individual with intellectual disability, higher yield with comorbidities, which is congenital heart disease and intellectual disability is 55%. So if you have a child who is intellectually disabled and have comorbidities, congenital hereditary disease, for example, your yield of this test can be 55%, right? So that's a very powerful technology for prenatal diagnosis, for example, for newborn with congenital anomalies and so on. And it should be used, and it is a little expensive than chromosomes, but it is, I know when I visited Aga Khan last time, they have this machine, they are working on it, and I hope they have started testing for that also. So, but again, now you are orbiting around the moon's orbit, and you have not landed on the moon. So you wanted to look at each and every rock and see is how they compare to each other and if they are changing with time and so on and so on. So that is called a sequencing technology, human genome sequencing. And let's go there, what sequencing mean. And you basically, as an astronaut, land on the ski and pick up any rock you want to pick up. So how sequencing work? It is like double-stranded DNA. You shred double-stranded DNA into small pieces and then denature it. Use your primer sets and amplify it. When you have abundance of amplified piece, you can analyze it by many different methods like capillary electrophoresis and so on. And that is when you're really on the moon. So sequencing technology have many different use and names, and or I'm just remember that this is an overview, so I'm not distinguishing between Sanger sequencing and whole genome sequencing or next-gen sequencing. Sequencing means all of this. So single gene sequencing, you can do it with Sanger sequencing. Multi-gene panel, you can also do it with Sanger sequencing or with NGS. Whole exome sequencing mean you just sequence every exome in the human genome, but you're not sequencing the introns. And the whole genome sequencing is when you screen the whole intron and genome. And I hope that's the ultimate technology, but I'm sure there'll be more technologies coming after that. So for example, cystic fibrosis, you can do it with single gene sequencing. Multi-gene panels, for example, cardiomyopathy panel, and neurological panel, or many different type of panel, myeloid panel in cancer, and lymphoid panel in cancer, solid tumor panels, and so on. So whole exome sequencing, for example, you are sequencing each and every exome of approximately 23,000 genes. And that is amazing thing to do. But the next is when you are really have a very high traveling plans, and you are on the moon, and you have a bike or a car which can drive you around it and look at each and every exome and each and every intron. And that is the ultimate sequencing is, and many places have started this. It's called sequencing intron and exomes of 23,000, approximately 23,000 genes, or it's called whole genome sequencing. It's expensive at present, but things are coming down. So because of this sequencing technology, and this is close to my end, what happens that many new applications come around. And I do not have time to go over in many details, but this is a very simple and very important and a game changer in prenatal diagnosis, for example. This is called non-invasive prenatal testing, NIPT. So when a pregnant woman used to come, or still comes in Pakistan or in many parts of the world, what you either do is a biochemical analysis, say that you have a 60% chance or 70% chance of having an abnormal child with Down syndrome or trisomy 18. That's not enough for one person. So if I am pregnant, if you tell me that you have a 60% chance or 70% chance of Down syndrome, and you have a 30% chance not having anything. So it's a very, very difficult and very troublesome question or answer. So then what they do is they used to draw amniotic fluid, and still drawing it, or CVS sample, and then culture them like we do with chromosome analysis, fish them with, say, probes for 21, 18, 13, and so on, or do microarray CGH to look at the whole genome. But that all involves invasive technologies. And invasive technology, there is a certain chance, low chance, it all depends on the operator, but there is a chance for up to 1% of losing that pregnancy. So when you go and talk to a woman and say that, oh, by the way, I have a test I can do, which will be a little more diagnostic, but you have a chance of 1% chance of losing your pregnancy. And that is also not really acceptable for many of us. So what we know that there is a placental barrier between child and mother. And through that barrier, there are fetal cells which go into maternal blood. So very smart scientists figured out that in maternal blood, you have maternal cells, you have fetal cells, and you have DNA, cell-free DNA, and you have a cell-free RNA and some protein also. If you start looking at it, if you start looking at fetal cells and somehow isolate these fetal cells, you should be able to look at that, sequence them, and see if that your target is present or absent. So all these prenatal diagnosis is coming down with NIPT. And Aisha and Myla and others have used this many times or every woman in US has actually offered this. And I think there is a company they have worked out details with, you can get it done in $100 only, right? $99 or $100. So that is a test for all prenatal pregnancies, high-risk pregnancies, and $100 means 60,000 rupees for Pakistan, for example, which if you look at other tests which are more invasive and cost a lot more. So what NIPT can do is screen for method of determining the risk that the fetus will be born with certain genetic abnormalities. Analyzing small fetal fragments of DNA circulated in pregnant women's blood. What type of things we can look at? Chromosomal disorders, microdeletion syndromes, and now we are beginning to use tests for single gene variants in fetal cells. And that technology is already in progress and should be common soon. So this is, in the end, what we say, if we have a big stuff, we use chromosome, we use FISH to analyze it. If we are looking at minor changes, we have sequencing technology to look at. And what are the benefits of genetic testing? Results provide a sense of relief from uncertainty and help people make an informed decision about managing their health, right? If I'm a female, if with a genetic test, I've been told you have a 90% chance to develop breast cancer, for example, if you have a certain mutation in BRCA1 and 2 gene in lifetime, it's up to me what decision I want to make. Do I want to live with that risk or do I want to take care of that risk? Same thing is colonic cancer, same thing is many other diseases. Negative result can eliminate the need of unnecessary checkups and screening tests in some cases. Positive result direct a person towards available prevention, monitoring, and treatment options like newborn screening, which is a whole different story. So these are just two examples. Poor feeding. It's a very common finding in young adults, young child, that my kid is not able to feed properly. And in that case, if you do a chromosome analysis or FISH analysis, many of these kids with certain features, which I'm not an expert with, but you can have a Wolff-Hirschhorn syndrome type of finding, or at least you can eliminate the poor feeding is because of one or the other reason. Then the second example is pediatric cancer. In West, pediatric cancer, most common pediatric cancer, which is ALL, is treatable, and these kids are having kids now. It's 95% of these kids will not die. But in Pakistan, when we visited in 2006 and 7, they were like, not even 30-40% of the kids were surviving, because reason was this genetic markers. So it depends what type of cancer it is and what do you see. If it's a hyperdiploidy, means you have more than 50 chromosome, you treat that cancer differently. And if you have a BCR-able translocation in ALL, then you treat it differently. The survival is a day-night difference between these two. So that's how the treatment is different between these type of things. And if you don't know what your ALL child with newly diagnosed ALL has, then you don't know how to treat it, period. So that was usually done by chromosome and fish analysis, and still can be done by chromosome and fish analysis. And that's why we, as a group with the Rotary Pakistan and APNA, Association of Pakistani Physicians of North America, we started a public sector lab at NICH, which is still working. And so in the end, I would say, what is the future of molecular medicine or genomic medicine, I would say, or it is high-level diagnostic, what we try to do, gene-level diagnostic, gene-level therapeutics. Right therapies delivered to the right person is likely to be one of the most important themes of healthcare in future. And the take-home message, and that's the last slide, biology, you are all biologists, right? Biology and very foundation of genomics teaches us that successful organisms are successful because they do one thing well, they adopt. And dinosaurs did not adopt, and see what happens to them. Cockroaches adopt very well, and they're doing great no matter what the circumstances are. So personalized medicine will not only help patients get better treatment, will reduce the disease burden and healthcare costs. Because at the time, I think I would like to end here, and I have a few more slides as maybe another lecture sometime, like what preventive medicine, why we call lab genetics, someone like us call lab genetic as preventive medicine. But it will definitely reduce the disease burden and healthcare cost in population. And thank you very much to Aisha and everyone for attending this, and I'm ready to answer any question.
We have some wonderful questions, but before I get to the questions, I just wanted to say again, thank you so much. This was such a lovely trip to the moon, and one of the participants really said it so well. They wanted to thank you to the moon and back, which was so apt for this conversation. So yeah, we have some really great questions from the chat boxes, and I would like to ask you those questions now. From Dr. Abrar Sheikh, he wanted to ask what could be the genetic makeup of cloned individuals. I think it could be a very simplistic answer.
So that's a very interesting question. So when I was at Baylor College of Medicine, we cloned cows, and we tried to see if immunoglobulin genes, human immunoglobulin, which is a very difficult gene to make and to have, but it's very important in many diseases. So we said we will clone the cow and have human extra chromosome in cow and see if we can get immunoglobulin. So what happens that expression is very different than the genome, for example. So you may have, so that cow, which is called Dolly, if you go and look at with my name, you will find that paper was published in Nature Genetics. And the cow has the extra human chromosome, but it was not expressing human immunoglobulin. So,
sir, excuse me, what was the fate of that cow? May I know?
Dolly,
what was the fate of that
Dolly.
cow? No, fate. What did happen in the
we
laws?
don't know. It means, it lives very happily ever after. We never kill those cows, but or any cloned animal. So there are many cloned animals now available, and they live happily. But the expression part of genomes, we don't know how will they express.
Respected sir, may I know about, I've heard about in some magazines about human cloning. I don't know further, but will you help me regarding knowledge of this, that anything regarding the human cloning?
So technically speaking, is it possible to do it? Of course it is possible to do it. Ethically speaking, is that happen ever again? No one knows. We haven't seen any.
Right, sir.
Okay, wonderful. Our next question came from an unknown user. They're using their Hawaii telephone. Anyways, their question is,
Hawaii is banned in US now. You know that.
That's probably why I don't know how to pronounce their name. Okay, here we go. What do you think are the biggest challenges for the adaptation of whole genome sequencing in Pakistan? Is it simply a matter of cost? With the population, there are likely many rare variants in both coding and non-coding regions, which may result in patients being misdiagnosed and having no course of therapy or support. So could you shed some light on that, please?
So a very good question, as a matter of fact.
Yeah.
And all of this, right? Of course, it is expensive. Of course, it's difficult to do. Like getting a pure water, purified water in Pakistan is, I come to Pakistan every day, every year, for example, used to before COVID. And I've seen many places have NGS type of technologies, like Armed Forces Institute of Pathology, like Aga Khan Universities, and many others. But they are not using it as it should be used because of all these reasons. It is expensive. It is finding something you don't know and you don't know what to do with that finding. It needs very high bioinformatician type of people who can analyze these small pieces of DNA. It's like computer programming, and with looking at A, B, C, D, A, T, C, T, or so on and so on. So all of this, but I believe that like we used to have, when I was in Pakistan, we used to have landlines of telephone, right? So landline of telephone was so difficult to get. You have to apply for an application, and then they set up some time in 2 years or 3 years that they will come and connect that landline to you. And then suddenly comes the wireless technology. So no one really cares about landlines anymore. So same thing is about genetic technology, that things will resolve by themselves. If with time, I think it's the cost is coming down, and the more important thing is that I think the electricity, the generators coming around, purified water or injectable water is coming around. So all these things matter, and getting access to these probes and how to use it. Pakistan has a lot of technology, but not many people know how to use it also. And that's the problem. I think a soft answer is everything what you mentioned.
Yep. Excellent. The next question is from Dr. Saqib Mahmood. He would like to know, will more advanced molecular technologies like NGS replace conventional cytogenetics and microarrays in the near future?
So again, Dr. Saqib can ask this question because he knows the answer. And what I mean is this, that so there, for example, loss and gain of chromosomal material. It used to be done by chromosome analysis or FISH analysis or array CGH analysis, right? But now with whole genome and whole exome sequencing, you can do copy number analysis also. But you need a very good bioinformatic team who can analyze that data. So people said that cytogenetic will be replaced 30 years ago, 20 years ago, when they start saying FISH and microarray, but cytogenetic still has its value, especially looking at balanced chromosomal rearrangements. And so far, next-gen technology is not able to completely analyze what you call balanced chromosomal rearrangements or mosaicism, for example. When you have more than one cell lines, where chromosomes you can look at and you can say that they are this primary line and the secondary line and the tertiary lines of clonality, for example. But RSCGH and NGS is a one shot of one time and place, and you see it if it's present or not present.
Excellent. Our next question is from Amir Niazi, and they would like to know, in Pakistan, cousin marriages are increasingly leading to genetic abnormalities among kids. Which genomic testing should be offered to cousins that are married?
So that's correct. Answer for you, question for you, I guess. So I will try, but what it is really is autosomal recessive diseases, for example. Right? You have to have both parents carrier of that disorder. So they don't have any physical features or symptoms of that disorder, but they carry on one of the two chromosomes, they carry a defective gene. And there is a 1 in 4 chance that child may be affected. 1 in 4 chance child will not gain the defective chromosome, and 50% chance that child can be carrier of the same disease as parents are. So those type of screening, there are screening panels, the screening technologies which you use with sequencing, and you can use for, depending if you know what your parents are carrying, that's very easy to screen while woman is pregnant, and that's the best ways to do it. So you decide, you know, very early in pregnancy if child is carrying what form of that disease child is carrying. Is he a carrier? Is he not affected totally, or he's affected? So I would say is screening parents first. In US, for example, Ashkenazi Jewish people, for example, they screen before they even marry. So they, before they date, their rabbi tell them to go and get the screening test. And if you're not fit for each other, find someone else.
Yes, very true. And it comes up in genetic counseling all the time. So this is our bread and butter, and nothing very unique to the Pakistani population. I can tell you that much. While it might be a little bit more prominent, but it's not unique at all. Okay, our next question comes from Amara Chaudhry, and they would like to know, are there any cures for genetic disorders besides counseling?
So cure is coming. Cystic fibrosis, for example, yes, there are some medications which are possible, and with some of the new genomic-based medicine, cure, for example, newborn screening can cure a lot of diseases. Or so lipid storage diseases or biochemical disorders, which are autosomal recessive, lysosomal storage disorders. If you detect it very early in life, there is a replacement therapy and your child will be fine. If you don't do it, the child will be depleted, disabled, and will have a lot of problem as we move forward. So treatment is not there for many of the diseases, majority of the diseases, but at least you have an option of the more treatment is coming around, and more than that, preventive measures are always there.
Mm-hmm. Yeah. Okay, so the next question is from Jolan, and this is more of a technical question. I am not sure how to say it, so I'm just going to read it out exactly how it says. They want to know, what is the difference between siRNA and mRNA in developing new drugs and vaccinations? Yeah, it's out of my scope, I figured.
Right. So I honestly, I cannot explain that also at present, but we will write her back when we know. What is the difference between different type of RNAs? That's a very technical question, and I do not know the answer at present.
Okay, yeah, good. Yeah, me neither. Okay, so next question is from Hamza Asghar, and they are interested in knowing, are there any methods exist for curing genetic diseases except for gene therapy and enzyme replacement therapy?
So prevention, the biggest treatment for genetic diseases are prevention, screening and prevention.
Yeah. All right, next question is from Bushra, and she would like to know, how far are we for the genetic testing of diabetes and cardiovascular diseases worldwide? Is it in use anywhere?
Yeah, that's a very good question also. So as you know that when you study genetics, you understand there are certain single gene disorders, there are certain multi-gene disorder, and there are certain multi-gene plus environmental disorders. So diabetes and cardiac diseases, they are very complex diseases, and they not only have multi-genic effect, but also have a lot to do with environment and place where you live and how to interact with genes and environment. So it is a complex process, but it is multi-genic and environmental interaction together.
Yeah, I could just add a little bit more to that. So there are polygenic risk scores that are currently being developed, but they're mostly in cancer settings, especially breast cancer settings. Unfortunately, they are quite controversial, as you might imagine, because these tests are being based off of genome-wide association studies. And as we all know, genome-wide association studies are done primarily 90+% of the time on the Caucasian population, and they do not provide any real significant data on any other population. Some of it has been done on the Chinese population, and even less has been done on population of African and South Asian ancestries. So it's quite controversial, and I would say long ways, if I could just say it in few words. Okay, next question is from Ansar Rabia, and she would like to know, what can you please comment on any drawbacks of whole exome sequencing compared to any of the previous techniques that have been in place?
Can you repeat that question?
Yeah, the next question is, can you please comment on any drawbacks of whole exome sequencing, comparing it with previous techniques?
So drawbacks, I don't see any drawbacks really, because if done right, it just looks at, so you all know that genes are more representative, or majority, or 99% of the genes are in exomes, not in introns, right? So that's why people think that exomes are actually are worth studying, and introns are actually called junk DNA. But junks can also have some, there are certain definitely disorders which are intronic disturbances or intronic deletions and duplications also. So I would say whole exome sequencing is at present is more refined and more acceptable. And the only difference thing I can see that if you're looking for a balanced chromosomal rearrangement, whole exome sequence may not be a good technology. But other than that, it is, that's the best technology there is at present.
Yeah. So some of the times when I think about this, I think about how sometimes where the phenotype is so complex and there's so many different diseases that could be in your differential, you're really not sure where to start. So you start by, you can do one of two things. You can either say, okay, let's do, these 5 single gene disorders that can add up the cost, or you could say, let's do, whole exome sequencing and see if we catch something. And it really could depend on, many things, the cost involved, the time that's of the essence, where's the patient and their needs, and where do we go. So quite a lot of things go into that. So that if you're looking at drawbacks, you could say that less trained, or not less trained, but when we don't know where to go, whole exome.
Right. So I would say just remember that hammer slides, right? Yeah. So you need to know which hammer you want to use for what condition and what situation.
Yeah. Okay. Our next question is from Mustafa. I'm going to butcher the last name, Mekare. And they wanted to know, what are your thoughts around PGD implementation in Pakistan and its ethical implications? And are the barriers more cultural or are they technological?
So PEG means pre-implantation genetics?
I would believe so. If Mustafa would like to unmute themselves and clarify, I think that they're talking about pre-implantation genetic diagnosis.
Right. So I see that
exactly
pre-implantation genetic is very important, right? So if I am carrying certain disorder, which is an autosomal recessive or autosomal dominant, and I don't want my child to be affected with that, and I have an option of in vitro fertilization and checking at pre-implantation genetics, of course, that's a very good choice. But doing it for sex selection, for example, like many people do it in India and many part of the world, that is very unethical, and hopefully it never happens in Pakistan.
Yeah, that's a great question, great answer, great response. The next question is from Farhan Bukhari, and he would like to know what is a good database for Pakistani rare disorders in terms
There is no database. There is no database for
there is no
Pakistan,
database
right?
for-
So we need to create one, and that's the
Yeah.
challenge. Hopefully, we will be dealing with how to develop population-based data for Pakistani population, which is again a very unique population.
Right. So, of course, a lot of you guys are probably very well aware of Genome AD. So I'm going to put the link in the chat for whoever is interested. They can go to this page and they can look for certain variants and see what the population prevalence is in various different populations. So you could, there is one for the, I believe there was a bunch of individuals from Lahore. So there is a Lahori, population data set there, but it's very limited, very small amount of individuals. And then they have one for Indian population as well. I believe they're Gujaratis from Houston, actually. So there's a huge Indian population in United States, and especially in Houston area. And they took some of those as individuals as reference for the Genome AD, and that there, you can find some information there. It's again, better than nothing, but it needs a lot more improvement. So if you're interested in going there, you can definitely try that website for references. Okay, next question is from Hadika Saeed, and she would like to know what is the future of next generation sequencing in Pakistan? What can be done in this regard, in your opinion?
We need to train more genetic counselors. We need to train more people who are bioinformatics, know bioinformatics. We need to train more people who correlate genotypic and phenotypic findings. And technology is technology. Anyone can learn, anyone can do it. But interpretation of that technology is where we are missing in Pakistan.
Yeah.
And that is one of our goals of PSMG, that to train more genetic counselors like Aisha and Myla, who can guide them what to do and how to do.
We're earnestly working on that goal as fast as we can. Next question is from Fatma Riaz, and she would like to know any thoughts you might have on diseases where epigenetics changes are implicated.
So epigenetics as I said, that you may have a very no change, very similar genome as a reference genome, but different genes expressed differently at different stages of life, at different environmental conditions, and different other factors involving also. It's very, very important in many cancer situation, and very important in, let's say, Prader-Willi and Angelman syndrome, which is basically a syndrome which is, if you inherit from father, you will have a different phenotype. If you inherit from mother, you will have a different phenotype. So it is very important, and is part of the whole genetic teaching training process.
Mm-hmm. Mm-hmm. Okay, next question is from Kifayat Hussain, and they would like to know, can telehealth play an important role to diagnosis of genetic diseases?
What can play a role?
Telehealth.
Telehealth, yes. So, Aisha can tell you she's doing all her genetic counseling by telehealth now. We are teaching this whole genome lecture series by telehealth now. So yes, of course, it can be very helpful. And that's what our goal is, to see how we can use telehealth to train and teach people and make more people who are well trained in genomics and genetic technologies and counseling.
Yeah, I would say so far we've been quite successful in establishing 2 telehealth clinics in Pakistan with the help of Dr. Syed Ejaz Ahmed, myself, Dr. Rizwan Naeem, Myla Ashfaq, Amreen Khan. There's a few of us that have started a couple of telegenetics clinics in Pakistan, and we've had some success in the counseling process. So far, we've only really had individuals that were already pre-diagnosed, and we were providing genetic counseling to some of their families and to clarify some of the concepts. These are really hard concepts, as you could imagine. Some of you probably would have to watch the recording that we did today to really clarify some of the concepts as is. So if learning individuals like yourselves can have difficulty understanding it, you can imagine how hard could it be for some of the individuals that don't have the privileges that you and I have. And so it's quite impressive on how much work is already happening in Pakistan. It's quite eye-opening for how much is needed. And so to Kifayat's point, telehealth is the future. That's where it all lies. We have to utilize the platform that we have to make the most of it. And the last question I would say is from Uruj and Amara, and they're both interested in bioinformatics and would like us to have some lectures on bioinformatics as well. I don't have any expertise in bioinformatics at all, so I would not want to comment on that. Do you have anything to say to that?
I would say they are very smart and very right that we should have some bioinformatician who is talking about, because Pakistan computer technology is very advanced, and if we will find someone who can give a lecture, so just put it down as one of the topic, and it will be like it will be continuing lecture series. So bioinformatics will be one of the lecture, and it's very important.
Thank you. Okay, so that concludes all of our questions that came in the chat box. I think we have a couple more minutes. Are there any other questions or thoughts anybody has for Rizwan or Mila or myself? We're happy to take them now. Okay, so we got one from Kashif Nisar. Their question is, what are the treatment and prevention options available to treat genetic disorders after diagnosis through these experiences, sophisticated genetic tests? Secondly, what are the methods of maintaining confidentiality of the genetic data as a result of genetic testing?
So fortunately, there's a very important part of whole genomic genetic spectrum, and we do have laws, we do have consents type of processes in US, and I hope when we teach this genetic counseling course in Pakistan, they will know more about it. But I'm sure that everyone in Pakistan is also very aware of confidentiality, and make sure you talk to your physician when you do that. And if you are a physician, to make sure that you are communicating with your physician, because it's a very important part of the whole genomic space. We've been taught that very aggressively that what people can lose job in US if they share or look for anyone's laboratory information, not only genomic but any medical conditions, for example. So yes, that's a very important topic, and maybe Aisha can give another lecture on consent issues in genetics, because minor and adults with mental disabilities, and they're very, very, very touchy and very important subjects which we need to deal with.
Yeah, I would second that. I would say one of the biggest things that we are concerned about in terms of telehealth is the concept of privacy, because we know that when we're over here on the computer and the patient is over there on the computer, you don't know what that environment is like. You don't know who else is in the room and who else can hear this conversation. And it really is sort of tricky, and we really have to align with the cultural concepts. Of course, if there's a family member who is really keen on being in the room with the patient, and that's their support system, you have to acknowledge that and realize that that's important just as much as privacy concerns, but then also allow the practitioners from our side, from the genetic side, to say, hey, I would like to talk to the patient alone, or whatever we can do to really give that sort of comfort level to the patient. We always have to walk on that tightrope every once in a while. Okay, so next question is about CRISPR, and they would this person, this participant, they don't have their name in there, but they would like to know how has CRISPR helped with hemoglobinopathy treatments?
So again, that's a very different lecture, and we should talk about CRISPR also. I believe that CRISPR is a very good technologies which can be used for many genetic disorders and drug development, as a matter of fact. So, but there is not enough time to really talk about. If you want to have my opinion, CRISPR technology will be more common, is becoming part of mainline medicine, and used in drug developments at present, and vaccine development, for example, and should be used more in inherited genetic diseases also.
Okay, next question we have is from Kifayat Hussein again. He's quite excited about this conversation, it seems like, which is wonderful. And the next question is, what is the responsibility of the government that provide a good and suitable environment as well as facilities so that we can aware about genetic diseases and minimize risk in future and solve the current problem? None of us are government people. We don't know anyone in the government. I don't know anyone in the government, but I'm sure Rizwan can shed some light on that.
So I did not get that question again.
Yeah,
Sorry.
it was sort of written in a way that I couldn't read so properly, but I guess what I was trying to say is, could the government play a role in creating facilities or create awareness about these genetic diseases by probably doing some PSA announcements or something along those lines? Is what I'm getting from the questions. Please correct me, Kifayat, if I'm wrong.
I think definitely government has a role to play, but government should not be moderating what to do and how to do type of thing. So it's a free knowledge based and an ethically important area which should be dealt with in the private sectors rather than government sector. Government sectors always, as far as announcement, PSA, public announcement, yes, that can be, that government can help and message. For example, very simple thing, as you can see, the message about prevention of COVID. Many different people have different opinion, but I think government has played a big role in forwarding that message and working with them. I think, and I hope thalassemia and most of the common things, preventable things, should be government announcement on these issues.
Okay, wonderful. So with that, we will say thank you once again to Dr. Rizwan Naeem for this wonderful discussion and a wonderful talk. He is our social butterfly. He's the reason why we had close to 100 participants today. And we rely upon your very good social skills very many times for your guidance and continued help and support. Again, I would like to say thank you to all the participants for taking the time on your Sunday evenings to have a conversation with us about this.
But before you go, what I want to say, please fill out that Google form. And if you like the series, please let other people know who might be interested, because our simple goal is to spread the knowledge.
Yeah, it sounds like some of you are having difficulty with the questionnaire. You can always email us by going to www.psmg.org, and we will send you a direct link so you can fill it out there as well. But it should be working. There shouldn't be any problems. I'll put it in the chat box one more time. Okay, wonderful. Thank you all, and have a lovely Sunday evening.
Thank you, everyone.
Khudafis.
Khudafis.